Thank you, Scott, and thank you to all of the speakers. Because of time constraints, we are going to bring all of the speakers back at the end of the presentations and open it up then for Q&A. I would encourage all speakers as we go forward to try to keep, I know it's an impossible task, but we still want you to try to keep within 10 to 12 minutes. The next panel is going to be on disease pathogenesis and consequences of infection. We have three speakers, one who will deliver his remarks here and the other two remotely. The first speaker will be Dr. Richard Kuhn, Professor and Head, Department of Biological Sciences, Director, Purdue Institute for Inflammation, Immunology, and Infectious Diseases, Purdue University. The second speaker will be Michael Diamond, Professor, Departments of Medicine, Molecular Microbiology, Pathology, and Immunology, Head of the Division of Infectious Diseases and Vaccine Development, Center for Human Immunology and Immunotherapy Programs, Washington University School of Medicine. And the third speaker will be Dr. William Britt, the Charles Alfred Professor of Pediatrics at the University of Alabama School of Medicine. Dr. Kuhn. Thank you.
Thank you, Mary.
Oh, and it's right
Got
up
it.
here. Okay.
Am I advancing these things? Next slide. Okay, so Zika virus is a member of the Flavivirus family of RNA viruses, and that's the good news because we know a lot about flaviviruses. Its genome is a positive strand of RNA, about 10,600 nucleotides in length, and as we've heard extensively, the virus is transmitted by mosquitoes. I'm going to talk today mostly about the structure of the virus, and the structure of the virus is going to presumably be similar to all of the other flaviviruses that we've seen. The virus particle contains a lipid bilayer membrane, which comes from the host. Inside that lipid bilayer membrane is a single genome RNA, together with many copies of a capsid protein. There are two other proteins that are found embedded on the outside, embedded in the lipid bilayer and found on the outside, and that is the envelope protein and the membrane protein. Next slide. This is a life cycle of flaviviruses, and we expect Zika virus to be very similar. The virus will bind to a receptor, be internalized, and inside in an endosome under low pH conditions, its membrane will fuse with the host and release the genome RNA, which is then infectious. It becomes translated. You make viral proteins. Those proteins establish replication factories for producing viral RNA and subsequently for producing new virus particles. Those virus particles bud into the endoplasmic reticulum. It undergoes a maturation process in which this immature, non-infectious virus becomes mature, and finally it's released from that infected cell. Next slide, please. Over the last 15 years or so, there's been a lot of work into the structure and function of flavivirus proteins, and so we have structures for many of the proteins of the virus. There are three structural proteins that I'm going to talk about today that compose the virus particle itself, and there are 7 proteins that are involved in interacting with the host and making new copies of RNA and new virus particles. Next slide, please. So the major surface protein on the outside of the virus is the envelope protein. This protein has 2 major functions. The first function is to bind to a target receptor. This is how the virus hones to specific cells within the body. Once it gets onto the receptor and gets internalized under low pH conditions, the E protein rearranges itself and carries out fusion of the viral membrane and the cellular membrane, and this releases the genome RNA into the cell. That surface protein, that envelope protein, is the major target for antibodies. However, we do know that in dengue virus infection, patients make antibodies against the M as well as the PRM protein, and we believe that this is important in virus biology. So what is seen here are 2 molecules of this surface protein. This is an E protein dimer. 3 domains of the protein are shown in red and in yellow and in blue. This is the part that is on the outside of the virus. There is actually another component to this protein that is embedded in the membrane, as not shown in this structure. Next slide, please. So that was the structure from dengue virus. We have the number of Flavivirus E protein structures. Unfortunately, we don't have it for Zika virus yet. But if we look at just a schematic comparing Zika virus with the other flaviviruses, we can note a number of similarities in terms of the organization, and we expect the E protein to be very similar in its structure. But there are some notable differences also. There are some insertions and deletions relative to the other Flavivirus E proteins. The glycosylation pattern is slightly different on Zika relative to the other flaviviruses. Next slide, please. So this is just showing amino acid identity amongst the structural proteins PRM and E and some of the replication proteins NS3 and NS5 with Zika virus. And one thing that is perhaps striking is that there is fairly high conservation in the replication proteins, less conservation when we look at the structural proteins, and in particular the PRM protein has the least identity among the different viruses. And so, although we know a lot about the flaviviruses, and in particular we know a lot about dengue virus and West Nile virus, there are clearly significant amino acid differences that are going to suggest that Zika is going to behave differently under certain conditions. Next slide, please. So many years ago, my colleague Michael Rossman and myself and members of our laboratories determined the structure of dengue virus using a technique called Cryo-Electron microscopy. This is a surface-shaded view of the virus, and you'll notice that the virus surface is extremely smooth. That E protein that I just mentioned, instead of standing up like we see with HIV or influenza, actually lies flat. It lies parallel to the surface of the virus particle. Next slide, please. So this structure, though, repeats this pattern of E proteins over and over again in a very ordered array, an icosahedral array. And so we can place, or we can see, the envelope protein in this structure. And on the next slide, we see the organization of that E protein that I showed a few slides ago. It's a very ordered arrangement on the virus particle. Next slide, please. We also know quite a bit from studies primarily of dengue virus of the maturation pathway of the flaviviruses, and we expect Zika to be very similar. We start off in the lumen of the endoplasmic reticulum with an immature, non-infectious virus particle, and that virus particle undergoes a lot of significant translational and conformational movements as it moves into a low pH environment in the trans-Golgi network. In that environment, a host protease cleaves the precursor protein PRM, resulting in now a mature virus particle. But one thing in particular is that we found with dengue virus, and to a lesser extent the other flaviviruses, that this host-controlled proteolytic event is not very efficient. And we believe that this inefficiency is actually important for the biology of the virus. Next slide, please. So, in fact, in many preparations coming out of both cell culture and from infected patients, we actually see a mixture of mature, fully infectious virus particles and what we call partially mature virus particles. This is the result of an inefficient cleavage. And in fact, in patients who are experiencing dengue and severe dengue infection, there are quite a number of antibodies that are directed against this PRM, this immature component of the virus particle. Next slide, please. So this is work that has been ongoing at Purdue between my lab and the laboratory of Michael Rossman. And what we see here are Zika virus that's been highly purified and has been cryo-frozen and prepared for electron microscopy. And I'll just say now that you can see that there are smooth particles in this preparation, and there are particles that are bumpy and that are presumably partially mature. Right now, we don't know exactly the breakdowns of those. We think the Zika virus is a little bit better at getting to be a mature virus than dengue virus, but we don't have complete data. I should, though, say that we've collected many thousands of particles right now, and we're closing in on a structure for Zika virus, and we should, I think, have that very shortly. Next slide, please. So just keep hitting it up. So this idea of maturity involves the amount of PRM and the host cleavage of this molecule. That influences the structure and another aspect that I didn't have time to talk about, the dynamics of the virus particle. These completely affect the ability of the virus to infect cells. They influence the entry. They also influence the exit of the virus from cells. This repertoire, this diversity of virus particles has a huge impact on the immune response, and also, we predict, has an effect on the disease and pathogenesis of the virus. Next slide, please. And keep hitting it. There's 3 points here. So here are unanswered questions on Zika virus structure. I just told you that the dengue virus population is pleomorphic. Different states of dengue exist due to variable PRM content. Temperature also plays a role, so what you might see in a mosquito is going to be different than what you might see in a feverish human. That influences the protein dynamics of the particle. So how does the structural heterogeneity of Zika contrast with dengue and the other flaviviruses? That's point one. Point two, are we going to see unforeseen surprises in the Zika structure, or is it going to be very similar to dengue and West Nile? I've already pointed out that we know that there are a number of differences in the structural proteins. So what unique features of these surface proteins are going to influence the antibody response? And if there are subtle or significant changes, how do they affect the ability to induce neutralizing antibodies, and how do they affect cellular receptors? And then finally, how does the structure of Zika influence receptor interactions and govern virus tropism? I think this is a critical question in terms of Zika virus compared with the other flaviviruses, because clearly Zika is getting into different cellular compartments that the other flaviviruses do not. And so finally, how will the structure inform function of receptor usage, tropism, and entry? And on my last slide, I just want to point out that there is a resource that people can go to look at genome sequences and protein sequences that are coming in from various Zika resources. This is the virus pathogen resource. This is supported by the National Institute for Allergy and Infectious Disease. And here's the website that you can go. It's a informatics platform, and I suggest that if you're interested, please go ahead and visit that. So thank you.